scientist at the Netherlands Institute for Neuroscience. Tycho completed his PhD at Baylor College of Medicine and then completed a postdoc at Princeton University. Throughout this time, Tycho's been really at the bleeding edge of open source optical and behavioral tool development. Most recently, Tycho developed the open source Miniscope, which allows multi-region recording and integrated optogenetic light delivery. Tycho also serves as associate editor for the open source journal Frontiers. He'll be talking today about the trade-off between function and size in the development of novel micro-endoscopes. So we're excited to have you. Thank you so much, Tycho.
Thanks so much for the invitation, Zach and Susie and Daniel, Denise and Tristan, of course, and all others that have been involved in bringing us together today. I think it's a great opportunity to exchange ideas about where we're headed with Miniscopes. And I'd like to take this opportunity today to expand a little bit on the design choices and also the trade-offs that have to be made when developing Miniscopes. So we truly had a Cambrian explosion of Miniscopes in the last decade. And of course, we have many variants of commercially available Miniscopes now, such as the Inscopics and Dork lens miniaturized microscopes, but several labs have started to develop their own. And for example, the Finch scope, which is really tailored towards imaging in birds and recording birdsong at the same time. More recently, we had a new optical design for a very lightweight microscope called the Featherscope, which was developed at MIT. We've had several multi-purpose open source miniscopes, such as the B4UCA miniscope and the Miniscope. And then also microscopes that have been more geared towards imaging from larger fields of view, such as the miniaturized mesoscope from Suhasa, and also the mini mesoscope developed at Boston University, the C-scope at Princeton University. And so there's a lot of these different miniscopes, and they're all sort of tailored towards specific scientific problems, but all of them have to make trade-offs in their design, be it, for example, the ease of assembly, the cost, dimensions, weight sensitivity, spatial resolution, sampling rate, and also platform compatibility. So yeah, so that I want to use as an example the development of our own Miniscope, the Miniscope, to kind of like highlight the sort of changes that we had to make along the way in developing our own Miniscopes. And it's like always a balance between like, what do you want? What is the goal of developing this technology? What sort of research questions do you want to address with them? And what sort of trade-offs are you willing to make in the design? So the primary goal for developing Miniscope was actually to be able to record from two regions at the same time. So we wanted to be able to record from cerebellum, which we have been researching for a long time. But we're also venturing a little bit more out of the cerebellum and looking at how the cerebellum and other brain regions are synchronizing their activity. So we really wanted to know how's the cerebellum and the cortex synchronizing activity during unrestrained behavior. And so that necessitated a new kind of design, a very small and compact miniscope. And at the time that we were posing ourselves this question and we had this need, there weren't really any miniscopes out there that were allowing us to do this. So we designed it from scratch ourselves, but basing ourselves, of course, on already available open source technologies, such as the UCA Miniscope project. So yeah, we began also through the very pleasant interactions with all these people in the open source community. We were able to determine, okay, what are the best sensors to use in our Miniscope? So we talked with several people, including Daniel, and we decided to go for the Python 480 image sensor. At that time, it was the sensor that was good in terms of sensitivity. It was better than the predecessor sensors that had previously been implemented in other designs. And it would and we would be able to fit it into a relatively compact design. So our mechanical design was also tailored towards making something that could allow us to fit two microscopes on a single mouse. And so of course, we had to make it lightweight. So we developed a microscope that could fit on a mouse. It had a small footprint, but was also lightweight. Because we were using very small, like very high density electronics on our PCBs, we were also facing a limitation that was that if you would operate the sensor clock at a high frequency, it would also heat up the electronics too much, and that would also impair the image quality. So we decided to switch off the phase lock loop to lower the clock frequency of the sensor, and also to mitigate this problem of overheating of our sensor. And so 30 hertz is still pretty reasonable for calcium imaging experiments, so we were fine to go with that option. And so in addition to having this small compact set of PCBs that we used, we decided to stack the two PCBs so that we had more space to add additional functionality. And one of the things that we added was, for example, G-sensor. And we thought it would be useful to have like quick readout of behavior without having to go through like very advanced behavioral tracking using, for example, machine learning approaches. And the other thing was that, if we can look at two regions at the same time with two miniscopes, why can't we just manipulate one region and also image in another? So that made us to also integrate a high precision LED driver that allowed optogenetic actuation of neurons in one remote region from the imaging site. Another consideration for us was that we wanted to have this cross-platform, the software to control the miniscope. So we wrote code that could be installed on any kind of laptop that you would have lying around, whether it was running Linux, Windows, or Mac. And one of the other design choices that was really necessitated for a dual side imaging miniscope was that we used the manual focus. Electronic focusing would be useful and could be implemented. We actually tested the electrowetting lens as well, but it added another additional half a gram of weight, and so we didn't want that. So we decided to forgo this and stick with manual focusing. And we also stuck with GRIN lens optics. At that time, we had established all our surgical procedures, so for us it was just convenient to stick with what we had. Of course, having a stack of lenses that is integrated into the microscope is maybe more durable and cost effective in the long run, and you're seeing that now being implemented also in the more recent miniscope technologies that are coming on the market. So in terms of the integration of our accelerometer in the Miniscope, we thought it was useful for simple behaviors, for example, turning. So there were some experiments performed by Bastien van de Boom in the lab of Ingo Willen at the NIN, Netherlands Institute for Neuroscience, and he looked at neurons in their dorsal striatum. So these neurons appear to be responsive when animals are making a turn. And so a turn contralateral to the area image, that's what these cells were responding to most. So in this case, we were imaging from right dorsal striatum. When animals were making a left turn, these neurons were also becoming activated. And we could use the G-sensor readout to really look at the left turns and the right turns, and then trigger off of those simple behaviors to establish that this wasn't indeed happening. So one of the trade-offs was with the accelerometer readout that we could only sample at about up to 90 hertz, because we're also reading out all the image data that has to also be transferred to the computer. So we had to interleave this data with the G-sensor data with image data. So that could be considered one drawback. But the benefit, of course, is that we had a very simple readout of the behavior. The addition of the high precision LED driver for optogenetic actuation was useful because we wanted to study this interaction between cerebellum and cortex. And so we had an integrated LED driver that could be controlled from the Miniscope software. So we could really easily synchronize the imaging data and know when we triggered the stimulation. And so we used that to look at what was happening to cortical activity when we stimulated cerebellum. And we observed this ramping activity in the cortical neurons whenever there was repeated stimulation in the cerebellum. And so because we were setting we were using a low power mode for the low frame rate acquisition or the 30 Hertz acquisition, we could also draw more current for this optogenetic stimulation lab probe. And that was an advantage, even though we didn't have the ability to acquire frames at a bit higher frame rates that this sensor is capable of. So, so we're quite content actually with the overall functionality of Miniscope, and that's quite small, and it achieved our goal of like being able to record from 2 regions that we were interested in. But we've also tried to pursue a different route, which is to see, can we extend the frame rate higher of our regular Miniscope. So we didn't have a lot of time and money to invest in like a big redesign. So we decided to stick with the sensor that we had, which had relatively large pixels, but we had some questions about whether it would be sufficiently sensitive to also use for high frame rate acquisition using voltage sensors. At the same time, we had the design that was relatively small and compact, so we liked that and we wanted to stick with that. And in order to go to these high frame rate acquisitions, we also needed to take care of the issue of heat dissipation because we had to switch the clock frequency of the image sensor to a higher frequency, which obviously led to a heating up of the image sensor with degradation of quality. And so we thought of a solution to that, and we decided to use air cooling. So what we did was we removed the enclosure of the sensor. We exposed both the PCBs that were part of the original Miniscope to air. And then we had a little tubing that allowed us to blow pressurized air onto the image sensor to cool it down. And so in this way, we could really go to a higher frame rate acquisition, up to 500 Hertz, also using the approach that Caleb demonstrated earlier, by selecting a region of interest, and I'll show some data in a minute of that. But the trade-offs here in this design were that given that we are drawing more current for the higher frame rate acquisition, we couldn't use as much current anymore to drive an optogenetic LED probe. And the other thing was at the high frequency acquisitions also, the G sensor data couldn't be written away anymore. So we're limited to combining G-sensor readout and 120 Hertz maximum frame rate for that purpose. Another thing that we encountered in this whole design process was that on Windows, we found a lot of frame drops when acquiring data. So we tested on Linux, and it seemed to work really well there. So this was actually like going a step back from our original idea of like we wanted to make it cross-platform, but Linux proved to be the operating system that worked best. Okay, so these are some examples of data that we acquired with our high frame rate iteration of Miniscope. So we acquired at 30, 120, 400 Hertz, and 500 Hertz, you see here on the left, from Purkinje cell dendrites that were transduced with GCAM6F. And so there's options in the software to either select custom size regions or to bin frames to combine pixels to increase sensitivity. But we're overall pretty pleased that even with this Python 480 sensor, we could still obtain reasonable data, even at high several hundreds of Hertz acquisition rates. And also in layer 2-3 cortex, we did some imaging of activity in cortical neurons. And also there we could acquire in bin mode up to 400 Hertz and still obtain pretty reliable signals. So you can see here some segmented Purkinje cell dendrites, and like the colors represent the segmented neurons and the signals that we obtained from them. And then on the bottom, you see the expanded traces, and they show that there's like a nice overlap of these individual transients that are predominantly triggered by complex spikes, although not exclusively, but predominantly. You see there's a nice overlap, and we can also start to distinguish these individual peaks much better than we would if you would acquire at a lower frame rate. And so here you see an overview of acquisitions at 30 Hertz, 120 Hertz, and 400 Hertz. And again, you see that at these lower frame rates, it's really hard to resolve individual peaks. But if you go to frame rates of like several hundred Hertz, you start to see more bursty activity or peaks that you wouldn't pick up otherwise. And if you look at the normalized transients, so these are spontaneous transients that were triggered off their onset at the various sampling frequencies, you see that at 30 Hertz, you know, it's harder to estimate the actual rise time of your signal, and it gets much better if you go to the higher frame rates of 120 Hertz and higher. Now, of course, we were also tempted to see if we could push this further, even if we didn't have a back illuminated sensor within this particular microscope, where we could still use it to pick up some signal from voltage sensors. And so we teamed up with the lab of Thomas Knupfel and Chen Song to transduce AS3 voltage sensor protein that was developed in the lab of Michael Lin. So we targeted this to interneurons in the somatosensory cortex, and we were able to pick up population signals. So I wouldn't say that we are able to pick up any or resolve any individual neurons, as would be possible with the more sensitive sensor, like Caleb demonstrated before. But we do pick up signals at 60 Hertz, and even like at hundreds of Hertz, there is still some signal buried, even though it gets quite noisy. And you can also see that the amount of light that we collect at these higher sampling rates is much reduced. So that was sort of an overview of like the whole design process and also the trade-offs that we made. I just wanted to consider a few things, looking back at all the Miniscope development that has happened over the years. I think it's clear that every Miniscope addresses a specific need. In our case, it was being able to record from cerebellum cortex at the same time. But there's always trade-offs that are made in at least some aspects of the design. So you don't have a one Miniscope fits all. And it's good that we have this large diversity of Miniscopes now available to all of us. Another thing is that once a Miniscope is designed and it's widely disseminated to other labs, a lot of the components like a camera sensor or other components like accelerometers or whatnot, they're quickly outdated, or there's newer versions available. And I think that really necessitates modular designs that are also more durable, so where you can swap out components more easily. And I think Daniel is already making big strides in that direction. And I also think that open sourcing technology is really driving innovation now in the field. We see like a lot of cross-pollination. We were inspired by the Miniscope project at UCLA to implement part of their technology. And we are seeing now that other people are picking up some of our design choices that were working out well. So I think it's just really good to see this more intensive collaboration between colleagues and more coordination between the developers to also improve the interoperability of future Miniscopes. I hope that's going to happen. And I think this Miniscope workshop is really helping with that. So with that, I would like to thank all the people that have been part of this. There have been several people involved. The Mechatronics department at the Netherlands Institute for Neuroscience has been instrumental in developing the Miniscopes. The funders. So I would also like to thank the open source community. And if you have any questions about like technical questions, or if you're interested in purchasing a Miniscope, you can email us at miniscope@nin.kw.nl. Or you can, if you want to assemble one yourself, you can go to our GitHub websites. So thank you very much. I look forward to the other talks, and I look forward to any questions you might have.
Thanks, Tycho. So we have time for about one question, if anyone has a question.
I have a question, if nobody's asking, and
Yeah,
it's both for Tycho and for Caleb. Have you tried Voltron? So when we were doing analysis of voltage imaging data, we found that Voltron actually was the one that was giving the highest signal to noise ratio. Did you try that? Also with the strategy of sparse expression that they are using?
Yeah, no, I think that's absolutely that. I mean, we're going to see Voltron is, of course, the sort of like the gold standard for voltage sensors right now. I mean, it's also a chemogenetic sensor, right? So you have to inject through the orbital space. You have to inject an additional factor to combine it with the genetically expressed protein. That makes it a little bit more complicated. So it was just a matter of being practical for us. Not that we didn't want to use that particular approach, but it's definitely something to do. Now, I do think that in the future, there's going to be sensors that are going to be even more sensitive and better. But we're running against the limits of what we can detect. That's true. And of course, we're also using a sensor that's not really suited optimally yet for the voltage sensing. But I hope that with improved sensors, maybe we become within that range. So now we haven't tried. That's the short answer.
Just to add on to that, the systematic localization, I think, is very important. And we're also very excited. I think the positive going sensors, I think, will help a lot with SNR. But there's positron, and the short answer for our group is exactly the same. We got scared by the retroorbital injection.
Thank you.
All right. So for audience who still have questions, you can leave your question in the chat or go join the lunch breakout room to ask your question. So thanks, Tycho, again for sharing this fantastic open source tool.